Okay, so DNA replication, we have the leading strand and the lagging strand. And so first of all, we have the helicase here. I won't say, yeah, right here, that breaks off the H bonds going this way, and then so we get the leading strand and the lagging strand. So on the lagging strand, you have the RNA primase enzyme, and what this enzyme does is it adds a RNA primer. Now this is like a base made of RNA, just on that with complementary pairing of the lagging strand, and that's all it does. It adds like a template, and then after that we have DNA polymerase 3. So here, and what this does is it adds DNTPs in the direction 5' to 3'. So this way, and for example, okay, let's make it blue. So it adds DNTPs like so, which are deoxynucleoside triphosphates. And then we have another enzyme, oops, over here, called DNA polymerase 1, and this replaces the RNA primer with DNA. So let's try it like this. So in blue you have the RNA primer, and let's say in red it'll transform into DNA, and yeah, these things are still here, the DNTPs. So it's a bit messy so far, but then finally a final enzyme called DNA ligase seals up and breaks between DNA fragments. So it basically, then what you basically have here is a complementary DNA strand, not RNA, complementary to this one, the lagging strand, this one here. And so here is DNA ligase, and yeah, that's basically how you come up with DNA replication. So you have RNA primase adds the RNA primer, DNA polymerase 3 starts replication. Next to the primer adds the DNTPs, so these things. And then the DNA polymerase 1 replaces RNA primer with DNA, so that's why it's red, like DNA. And then the DNA ligase seals up between the DNA fragments, so it basically creates the covalent bonds that make the backbone of the DNA. And then for the leading strand, I'm not too sure about this. All I know is that basically the same thing happens, but I'm not too sure why it's called leading strand. And that's basically it.